Our next speaker is going to be Corday Selden. Corday, I'm just going to give a very brief introduction. Corday just finished her PhD at Old Dominion University, working on nitrogen fixation in the real world. And so she's going to talk a little bit about nitrogen fixation in the real world. Here you go.
Thank you very much, Paul. And thank you, Dennis. That was a really, really great talk. I enjoyed it a lot. All right, so now I want to switch gears a little bit for a moment from thinking about nitrogenase on a molecular level to thinking about nitrogen fixation on an environmental scale. And in particular, I would like to say just a few words, this is a short talk, about the role that diazotroph diversity, and specifically diversity in metabolism and ecology and physiology, plays in regulating the where and when nitrogen fixation occurs in the environment. And I'm specifically going to focus on marine systems, in part because I'm an oceanographer by training, but also because nitrogen fixation plays an essential role in the ocean by relieving nitrogen limitation of primary producers in the surface ocean, and thereby exerting an influence over global climate, right, by facilitating carbon export via the biological pump. So when we think about marine nitrogen fixers, those of you who have thought about marine nitrogen fixers probably think about cyanobacteria like the ones that I'm showing here, and specifically cyanobacteria that are active in very low nutrient regions of the ocean, right, predominantly the central basins. And this is because there's terrestrial sources of fixed inorganic nitrogen species like nitrate in coastal zones, right? And nitrate also accumulates at depth because of remineralization, right? We have net heterotrophy, right? So the organic matter that sinks out of the euphotic zone is regenerated. And you know, this fixed inorganic nitrogen is largely thought of as being more, less energetically costly to assimilate than N2 gas. And so for this reason, we think of nitrate, right, and ammonium as well as inhibiting nitrogen fixation. And this is certainly true, on a direct physiological level for organisms like the one that I'm showing here, which is called trichodesmium. But there's a whole host of, last thought. So that's a negative, the nitrate is a negative feedback. The positive feedbacks that we typically think of are wind-driven inputs of iron, right? Iron in the form of dust from the continents, phosphate from the deep sea, also from dust. And these are really the major factors that we think of as controlling the marine nitrogen inventory, right? Or at least controlling the inputs via N2 fixation, which is the major input for the inventory. But there's a whole host of other diazotrophs that we think are active, right? And we think this because of a lot of work that's been done surveying NIF-H, which is usually used as a diagnostic gene for diazotrophs in the environment. And these include eukaryote symbionts, which we frequently find in coastal zones, and has raised questions about nitrate inhibiting their activity, as well as non-cyanobacterial diazotrophs in the deep sea. And so I just want to make a few very general comments about these two groups and how thinking about their physiology and ecology can relate back to how we interpret, how we think about the feedback mechanisms involved in the nitrogen cycle and sort of the distribution of nitrogen fixation across different biomes. So these non-cyanobacterial diazotrophs are largely proteobacteria, presumably heterotrophic, and we find them throughout the deep sea. We find them in surface waters. They're just everywhere. But having the genetic potential to do something doesn't mean it's actively happening, of course, right? And when we look at the activity of these organisms, it's it appears to be very low or not present throughout a lot of the deep sea, but we do find it in low oxygen environments. And this kind of makes sense because nitrogenase is sensitive to oxygen inhibition, meaning that organisms need to invest energy in strategies to avoid oxygen inhibition when in oxygen environments, right? So that energetic cost is relieved when we're in a low oxygen. We being the proteobacteria are in a low oxygen environment. And these organisms, where they are fixing nitrogen, appear to be stimulated by organic carbon, right? Which again makes sense if they're heterotrophic. That being said, this input seems like, I would argue it's probably relatively small, but nonetheless may be important on local scales where local sources of iron can result in high rates of nitrogen fixation. In contrast, these eukaryote symbionts, when we find them on the continental shelf, while they are present and active in areas where there's terrestrial sources of fixed nitrogen, we find the highest rates, which can be very considerable, sort of just past this area where nitrate is really drawn down, for the most part, either by other primary producers or via denitrification in the sediments. That being said, there's been several studies suggesting that their activity may be driven by upwelling of deep nutrients, but even excluding phosphate being a potential factor promoting nitrogen fixation, there was a study out of the California coastal system suggesting that just nitrate on its own could stimulate nitrogen fixation by these organisms because of their complex symbiotic relationship with the host. All right, so my point here is not to just throw up a lot of arrows, but rather I want to leave you with this one message, which is that the ecology and metabolism of these microbial groups that are mediating these important biogeochemical processes really affect the distribution and the regulation of these processes, right? And so we need to incorporate it into our thinking about both feedback mechanisms, right, and the distribution of these processes across different biomes. And in my last minute here, I just want to make one more comment, and that's that of course, when we're dealing with a modern environment, there's a lot of tools at our disposal to investigate these biogeochemical processes and to investigate community metabolism. These things are much harder to do when we're looking back into the past, as I think this group is very well aware of. Often when we're thinking about nitrogen cycling in the past, we're looking at nitrogen isotopes. But another tool that we're interested in the context of ENIGMA that may be potentially very useful is looking at metal isotopes. So the metals that are retained in organic matter, right, in a cell that then ends up being organic matter in the rock record, is reflective of the activity that cell was doing. And in particular, when we look at the isotopes of the different metals in organic inclusions in the rock record, for example, but in any organic matter, including in the modern environment, right, we can get some information, some additional information about what that cell was doing. And this is because different biological processes fractionate metal isotopes. I'm throwing a lot of different processes here that are probably important, but the one that I just want to mention briefly is these boxes 4 and 5. So intracellular trafficking of metals, right, and their incorporation into enzymes that have somewhat different structures and binding sites can result in we believe can result in distinct metal isotope signatures for different metabolic processes. And this is true of nitrogen fixing organisms, of diisotopes, when they're fixing nitrogen versus when they're assimilating nitrate. When we look at their molybdenum and iron isotopic signatures, they're distinct under those two conditions, likely because of intracellular processes and not because of uptake. All right, so, and that was the citation for the comment I just made. All right, so I think I'm at time, and I'll just leave this slide up for a moment if we want to have any questions, or if we can just move on.
Thank you very much, Corday. So we are open for questions. We can continue this conversation later with the other questions. In the world of nitrogen fixation, Corday, I have one question which has been bothering me for some time, for many years actually, since it was proposed, I think, by Candace Oviatt at University of Rhode Island, that we had nitrogen fixers in the continental margins. Why would you spend all that energy fixing nitrogen if you have access to nitrate or ammonia, which is basically free?
Well, okay, here's a question for you. If you have lots of energy, why do you need to be picky? Right, I think the base assumption behind your question is that organisms are always going to be as efficient as possible. And I think that makes sense if you're in a system where, you know, your energy limited, right? If you're in the oligotrophic open ocean and you just don't have that many resources, then of course you're going to be as efficient as possible, right? If you're very poor, you're going to spend your money, you're going to you know, very tightly regulate how you spend your money. You know, in a coastal system, is that really the case? And in particular, if you're
in phosphorus, if I have the ingredients to make more of myself, most of that energy is spent in protein synthesis. So about 85% of the phosphorus in a cell are in ribosomes. And most of the ATP that is used is in protein synthesis. So if I can get ammonia or nitrate into a cell and I have phosphate, I mean, why not just grow faster? It's a funny, I don't expect you to have an answer. It's just
well, I have one more comment to make on that front. And that's as relates to the nature of the symbiosis. So if, you know, I think if we're thinking about a free living diazotroph, sure. If we're thinking about a symbiont whose carbon source is dependent on reciprocal nitrogen production, then, you know, that there's no situation where, you know, not producing nitrogen is an option. If you stop producing nitrogen, you stop getting carbon, right?
Yeah. So Azul is asking about nitrogen, the metal isotopes for ancient metabolic processes. He thinks it's a great idea. I think we'll save that for the discussion at the end of this sequence. And I think, you know, everybody likes likes various isotopes of metals. I will just caution people that the heavier the isotope is, the smaller the isotopic fractionation will be. So that's just a very simple rule from Harold Urey. And so if you're going to go into, for example, iron isotopes, we have huge isotopic fractionations in humans in our blood, depending on diseases, which we can measure. But when you start to get up to very high atomic masses of metals, this becomes a smaller and smaller fractionation problem. Be that as it may,